Hello everyone and welcome to this webinar, Great Science for the Smaller Things, Innovative Solutions for Streamlining Nanopharmaceutical Production. I'm Margot Salets, Marketing Officer at Gattefossé, and I will be the moderator for today's session. This webinar is co-organized with RCPE and Skyepharma, and both members of the NanoFacT consortium that our speakers will talk about later. Some technical precisions before we start. You can ask your questions in the chat throughout the presentation because we will have a Q&A session after the presentations. And please note that the session will be recorded and will be available on our website and our partners' websites upon request. So, without further ado, I am pleased to introduce our speakers. So from Gattefossé, we welcome Manon Rossano. She is project leader and a formulation scientist specialized in lipid-based systems, including emulsions, SEDs, and nanocarriers. And for the past 3 years at Gattefossé, she has been developing and optimizing lipid-based formulations to enhance performance. From RCPE, we welcome Dr. Caroline Tetishka. She is a senior scientist focusing on the development, manufacturing, and characterization of nano-drug delivery systems and the process technologies required to translate these systems into medications. She places a particular emphasis on lipid-based nanosystems and is actively involved in establishing continuous nano-manufacturing processes. She has led several projects in this field, including the NanoFacT project. From Skyepharma, we welcome Vanessa Bourgeot. She is currently Innovation and CMC Leader, driving the strategy and development of innovative oral drug delivery technologies. At Skyepharma, she brings a combined end-to-end vision of APIs and galenic formulation, supported by a strong chemical engineering background, enabling her to align API properties, formulation design, and industrial constraints from early development through CMC execution. And finally, still from Skyepharma, we welcome Valentine Darret, currently a PhD student. She works in collaboration with the LAGEP in France, and she is developing a platform of lipid nanoparticles to enhance the oral bioavailability of APIs that present low solubility and/or low bioavailability. So our speakers will take turns speaking following this agenda. First, Manon will present the advantages of using nanoparticles in drug delivery and which excipients to use. Then Caroline will present the NanoFacT consortium and how to scale up nanoparticle manufacturing using continuous manufacturing. And finally, Vanessa and Valentine will present a proof of concept with abiraterone before the Q&A session. So I now give the mic to Manon for the first part of the presentation.
Thank you, Margot. Hello, everyone, and thank you for joining us today. I'm glad to open this webinar dedicated to lipid-based nanoparticles and their applications in drug delivery. So as introduced, I'm part of the Pharmaceutical Application Laboratory at Gattefossé, where we focus on innovative lipid excipients and on helping formulators overcome complex drug delivery challenges. So in this first part, I will walk you through the key advantages of using nanoparticles in pharmaceutical formulations and explain which excipients you can rely on to design efficient, robust, and scalable systems. So let's start with the broader formulation context. So as you know, between 70 and 90% of new drug candidates are poorly soluble. Many belong to BCS class 2 or 4 with poor solubility, poor permeability. And these new drugs show increasing lipophilicity and molecular complexity. This characteristic leads to low aqueous solubility, poor oral bioavailability, instability issues, and overall difficulty in achieving consistent therapeutic exposure. So in this landscape, the formulation technologies are absolutely critical to unlock the full potential of these molecules. And among the different approaches, nanoparticles are emerging as one of promising solutions. So formulators have several tools in their toolbox. They can use salts, co-crystals, polymorph optimization. They can work with amorphous solid dispersions using polymer, cyclodextrin inclusion complexes also, and classical lipid-based formulation that we call LBF. However, these approaches are not always sufficient sometimes, especially for very lipophilic or high molecular weight or unstable drugs. So advanced system, and particularly nanoparticle system, can offer improved solubilization capacity, so with better stability and enhanced performance. So what are nanoparticles? The nanoparticles represent a broad family of different systems made from different materials. So it can be polymers, metals, lipids, but more. And they share the common goal: improve drug delivery efficiently and safely. In today's presentation, we will focus specifically on the lipid nanoparticle, which I will distinct also from liposomes, where the liposomes rely only on phospholipid bilayers, whereas the lipid nanoparticles are solid or semi-solid lipid matrices. And here, the lipid nanoparticles, you will see, combine the best characteristics from several points of view: the biocompatibility of the lipid, but also the structural versatility of the polymeric systems, and also the colloidal stability of the emulsion. So here we are looking at what we consider the first generation of the lipid nanoparticles. These are systems that are based on solid or mixed solid-liquid lipid matrices that are stabilized by surfactant. This means that we are not addressing the broader class of LNPs that are used for nucleic acid delivery, such as ionizable or charged lipids, pegylated lipids, or multi-component systems that have been developed for RNA vaccines, for example. These modern LNPs are different categories with different mechanisms and formulation constraints. So the lipid we are talking about here, these lipids are typically relying on either a solid lipid core or a mixture of solid and liquid lipids. So the solid lipid core offer high stability and strong protection of the API. They are called the SLN. And when you integrate some liquid into this core, you create the NLC. And this generally increases the drug loading capacity and reduce the drug expulsion during the storage. And all these systems, they are stabilized with surfactant, ensuring controlled particle size and colloidal stability. Overall, these lipid nanoparticles stand out for their biocompatibility, since their reliance on generally recognized as safe, and their ability to protect and deliver the sensitive molecules. So here you can see also what is happening when you add an API. So when you integrate the API into the lipid matrix, depending on its solubility profile and the affinity for the lipid, the API can be molecularly dispersed, located in amorphous clusters, or partially crystallized. So in SLN, you can generally have a lower drug load due to the high crystallinity of the symmetric brick wall model, and sometimes that can lead to drug expulsion during the long-term storage. While for NLC, a higher drug load is possible due to the nanostructure and leads to more physically stable system. So understanding this distribution is crucial because it influences the stability, the release behavior, and the overall performance of your nanoparticle. So the benefits, so why we combine, why combine lipids with nanoparticle technology? So because it brings multiple synergistic benefits for bioavailability. With the lipids, you can increase the drug solubility and maintain the solubilization throughout the digestion. We can reduce also the food effect, which is often problematic for lipophilic molecules. LBF can also, with some excipient, we can promote the lymphatic uptake, by bypassing the first pass metabolism. And they can also increase the intestinal permeability using permeation enhancer excipients. The nanostructure systems enable to protect the API from enzymatic degradation, and they reduce the efflux through transporters such as PGP, P-glycoprotein. So when we combine lipid excipient with nanostructure system, we obtain a platform capable of overcoming several hurdles at the same time. And another key advantage of the lipid nanoparticles is their versatility in terms of administration routes. So from topical to oral, parenteral, pulmonary, or mucosal delivery, for example, this system has been investigated across a wide range of therapeutic needs, and this gives the formulator the opportunity to design a technological platform that can be adapted to different application and dosage form. And the lipid nanoparticles can be then incorporated into nearly all the dosage forms, such as tablets, capsules, suspensions, solutions, topical, and even powders for reconstitution. So for oral solid dosage form, obviously the feasibility will depend on the manufacturing process selected, and to obtain a dry powder, whether for tablet, hot capsule, or sachet, the nanoparticle dispersion must be dried or the solvent evaporated through processes such as spray drying or fluid base coating, for example. But before diving into excipients, let me briefly introduce Gattefossé. We are an independent international company specializing in ingredients and formulation solutions for the beauty and healthcare industries. And in pharmaceuticals, we design and produce high-quality lipid excipients that support solubilization, bioavailability enhancement, and advanced drug delivery systems, such as nanoparticles. Our headquarters and first production site are located in France, next to Lyon. We operate 3 production sites in France, Singapore, and in Texas, in the US, as well as 4 technical centers of excellence around the world. And with 12 affiliates and more than 40 distributors, we serve the customers in over 19 countries, supported by a team of more than 300 employees. And the pharmaceutical division supports the customers across human health, animal health, nutraceutical application, and our lipid excipients are used across a wide range of administration routes, including oral, topical, transdermal, rectal, and vaginal application. And depending on the formulation needs, our product can act as solubilizer, bioenhancer, skin penetration enhancer, lubrication agent, or sustained release matrices. So, which Gattefossé lipid for nanocarrier? So we're going to focus here just on which excipients actually select when designing nanoparticle system. So for solid lipid nanoparticles, materials such as glycerol ester, like compritol 88880, glycerol 805, or even geleol, and also gelucir 4301, which is a hard fat, offer different melting range. And the melting point is a key selection here because it directly affects the manufacturing process. So high melting lipids may require elevated processing temperature, which can be sometimes problematic when working with thermosensitive API, but lower melting lipids may facilitate the process and also influence the internal structure of the nanoparticles. For the liquid lipids, material like capriol 90, Mazin CC, PCol, Labrafac, lipophil, they bring improved loading capacity and they help tune the internal structure of the NLC. And also, finally, the surfactants that we have in our portfolio, such as like Gelucir 4816 or Labrazole ALF, they can ensure the proper stabilization and allow the fine control of the colloidal system with the HLB value around 70. So you can also see among these excipients that the ones with the star are permeation enhancer, and they are based on esters of medium chain fatty acids. So by combining these excipients, the formulators can design tailored lipid matrices optimized to their active of interest. But of course, despite their potential, nanoparticle systems still face several challenges when moving from lab scale to industrial production. Today, for example, only a limited number of nanoparticle-based products have reached the market using these lipid technologies, which highlights the need for continued innovation to simplify the development, strengthen the process reliability, and facilitate broader industrial adoption. So scalability, reproducibility, process control, regulatory requirements must all be carefully addressed. And this is precisely why collaborative initiatives such as the NanoFacT consortium, for example, where we were working with the team here and that Caroline is going to present you, are essential to bring innovation all the way to industrial application and to the regulatory acceptance. So I hope this introduction has given you a clear understanding of why lipidic nanoparticles are relevant for the modern drug delivery and how lipid excipients can help you design effective systems. And I'm happy to transition to the next part of the webinar with Caroline from RCPE.
Thanks a lot, Manon, for this very nice introduction. It's a pleasure for me to welcome you all to our webinar. First of all, I would like to give you a short overview about RCPE. RCPE is called Research Center for Pharmaceutical Engineering. We are located in Graz in Austria, and we are global leaders in pharmaceutical process engineering. We're currently around 150 employees out of 21 nationalities and have more than 150 industrial and scientific partners worldwide. Since we are a research center, our currency is our publications. Since the beginning of RCPE, we now have around 700 papers and in-house equipment of roughly 19 million euros. We have around 800 square meters state-of-the-art facilities, a dedicated pilot plant, and can work with substances of up to OEB4, even highly potent substances. We have an extensive selection of different equipment in-house, from material science lab to analytical laboratories, pilot plant for solid state manufacturing, but also liquid dosage forms such as nanosystems can be prepared. Here you see a selection of our partners from pharma companies to equipment providers, material providers, and also international and national scientific partners. We're structured in 3 different areas. Area 1, science of quality, deals mainly with process simulation and modeling. Area 2, advanced formulations. They work mainly in the field of advanced dosage forms, physicochemical profiling, and biopharmaceutics. Area 3, next generation manufacturing, where I am working in, is divided in 2 departments. The CC flow department, which deals with API synthesis in flow, for example, or novel flow chemistry, and process and manufacturing science. We have a look on continuous manufacturing, process control, and PAT. In terms of nanoprocessing, we first of all always characterize our materials. We then select appropriate processes to prepare nanoparticles that are then subsequently characterized to assess their quality. We use the in-house facilities and the support of all the 3 areas, for example, to implement also predictive modeling, use the engineering know-how, or have a look on QPD and QPC. In terms of collaboration, we work in different streams. On the left-hand side, you can see the funded programs we work in national and international programs, as well as service projects. And then we also work in our K1 funding projects. These are specific types of funding through the funding of our research center. And this type of funding we applied in our so-called NanoFacT project that was called Robust, Flexible, Modular, and Continuous Nanomanufacturing Platform, and ran from June 2022 until June 2025. The partners of the project were the industrial partners Gattefossé, In-Process, LSP, Knauer, Microfluidics, Skyepharma, and a scientific partner we had, University of Graz, and we at RCPE were the project coordinators. So why do we want to work in the nanoparticle field? First of all, nanoparticle production in large scale is performed by 2 main approaches. This is the top-down approach, where we reduce the bulk size into the nanometer range, and the bottom-up approach, where self-assembly of the nanoparticles occurs from molecular level to the nanoparticles. In top-down, in the industry, usually wet media milling, high-pressure homogenization or microfluidizer processing is applied, whereas in bottom-up, we use, for example, impingement jet mixing, ethanol injection, or microfluidic processes. Thereby, it's important to maintain the characteristics from small-scale to large-scale production, as we see, especially in large-scale production, several challenges. On the one hand, in process on quality, we have high development and manufacturing costs. The processes are multi-step batch-based processes, and the manufacturing of the nanoparticles is very complex. In terms of efficacy and safety, we often see a non-reproducible production, which leads to inconsistent therapeutic performance, and we have no or hardly any quality control. That's why, as part of NanoFacT, we aimed to develop a continuous manufacturing line equipped with process analytical technology to achieve nanoparticles with desired properties. We selected in this project to work with lipid-based nanodrug delivery systems. So we selected 3 types. First of all, nanoemulsions composed of a liquid lipid and a stabilizer, solid lipid nanoparticles composed of a solid lipid and a stabilizer, and a mixture of both, the so-called nanostructured lipid carriers, having solid lipid, a liquid lipid, and a stabilizer. Throughout the project, we worked with the solid lipids Presirol Atto5 and Gelosir 4301. The liquid lipids we chose were Labrafac Lipophil and Capriol 90. As a stabilizer, we used Twin 80, and the model drug we used was Edaravone, a BCS class 4 drug with low solubility and low permeability. As top-down approach, we used the microfluidizer technology. In this, the sample is fed in a feeding chamber. Then, through a high pressure pump, it is pumped with high pressure into the interaction chamber, where the particle size reduction and the homogenization occurs. Generally, there are different chambers available. The Y chamber is typically used in lipid-based systems, whereas the Z chamber is more often used for the manufacturing of nanocrystals. The scale-up in this case occurs through parallelization of the channels in the interaction chamber and can be easily performed. As bottom-up approach, we selected impingement jet mixing. In this case, an anti-solvent and the solvent are mixed with high pressure pumps into an impingement jet mixer. There, the self-assembly of the nanoparticles occurs due to differences in saturation solubility, and subsequently the product can either be transferred to further post-processing steps, or we can also quench, for example, the product with a buffer or with water to dilute our sample. The scale-up in this case also occurs for parallelization, and as a PAT tool, we selected, in our case, specially resolved dynamic light scattering, which is a special form of dynamic light scattering where highly turbid samples can be measured in flow without dilution, whereas in classical dynamic light scattering, we need to dilute our samples to prevent multi-scattering, and also only static measurements can be conducted. The top-down line in the end was composed of a high shear mixing step directly connected to the microfluidizer processor and the cooling step. Since we worked above the melting point of our lipids, we implemented a temperature control to be able to work above the melting point. The PAT tool was implemented inline and online, but we also used it as offline characterization tool. In addition to that, we used at-line dynamic light scattering and also compared it with offline dynamic light scattering. Moreover, we also performed offline characterization, for example, using HPLC to determine the encapsulation efficiency and the loading capacity. Throughout the process, we had different physical states of the nanoformulations. Since we worked above the melting point of the lipids, the process temperatures were dependent on the applied lipids. So first of all, during the microfluidizer process and the high shear mixing process, we had liquid nano droplets, and then during the cooling, the recrystallization occurred, and after the cooling, we had solid nanoparticles. As you can see on the graph on the left-hand side, the particles or the nano droplets during manufacturing were generally smaller in size compared to the solid particles after the crystallization. And generally, we can also say that the Preserol nanoparticles were slightly smaller than the Gelocir nanoparticles. We then had a look on the influence of the cycle number while measuring different formulations using inline SRDLS. What we found is that the particle size decreased with increasing cycle number until it reached smaller sizes approximately after 6 cycles, which then reached also a plateau, and we optimized then our process in a way that we selected the optimized cycle number for all formulations. We subsequently then had a look also on the comparison of SRDLS and DLS. As mentioned before, the inline size measurements of SRDLS were generally smaller than the offline after the solidification of the nanoparticles, but we also saw differences when we worked with DLS and SRDLS. DLS generally provided smaller sizes than SRDLS. This can be related to the different light sources that were applied in the 2 devices, leading to a size difference of around 13 nanometers. Loading the formulations with edaravone, we saw that we achieved a higher encapsulation efficiency and loading capacity using nanoemulsions. This can be related to the fact that the drug had a higher solubility in the liquid lipids than in the solid lipids that we applied here. Now coming to the bottom-up line, as mentioned before, we worked with impingement jet mixing that was directly coupled to SRDLS to determine the size in real time. In this case, we had a specific focus on the development of a digital framework. This framework was developed in 2 stages. First of all, we had an automated DOE where we optimized our formulation and our process to achieve a target size of around 80 to 90 nanometers of our formulation that was composed of labrafac as lipid, using twin 80 as a stabilizer. In the second stage, we had a quality control algorithm where after manufacturing, we automatically detected the particle size, and the algorithm would then automatically see if our particles would go further via the valve to the product container, or we would have to put it into the waste container since we see a deviation. As you can see on this slide, we worked with different setups and compared the inline measurements with offline measurements. For example, runs A, B, C are stable runs with approximate sizes of 100 nanometers and good correlation between inline and offline DLS measurements. Whereas run D was a run where we saw deviations with polydispersed samples that would then go to the waste. Coming to the real-time quality control that we then implemented, we determined a target size and then performed included measurements. At the moment, the discharge would be on, meaning now our sample would go directly to the waste container, and we would adjust the pumps in a way that after a certain time period, it would go automatically again into the target size. In conclusion, we were able to establish a combined top-down and bottom-up manufacturing platform for precise production of nanoparticles. We implemented inline SRDLS for real-time size monitoring, enabling immediate process adjustments and consistent product quality. Using the top-down line, we were able to work under solvent-free and temperature-controlled conditions. Using the bottom-up line with the path-driven automation framework, we were able to rapidly optimize the settings. We could minimize the operator intervention and divert out-of-spec material. In addition to that, the platform is also broadly applicable to other nanodrug delivery systems and a wide range of drugs and multiple routes of administrations. With this, I would like to hand over to Vanessa and Valentine from Skyepharma, who will introduce Skyepharma and give an overview about use cases performed in their facilities.
Thank you very much, Caroline, and hello everybody. Thank you for joining this webinar. So first of all, let me present Skyepharma in a few words. So we are a French CDMO, so Contract Development Manufacturing Organization. So we are based in Lyon, in France, near the airport, and our focus is the manufacturing and the packaging of oral solid dosage forms. So we cover a broad range of activities accompanying the pharma player from preclinical stage up to the commercial manufacturing post-market approval. So some few figures. Last year, we had over 30 million euro of turnover. We have a facility of 22,000 square meters over a piece of land of 65,000 square meters. The company counts over 220 employees, including 25% of them working in R&D. So as a CDMO, we offer standard services in the field of oral galenic development, so including the development of formulation from scratch, but also we are manufacturing and analyzing lab-scale prototype. We are able to do the scale-up and clinical trial material, performing the industrial manufacturing as well as commercial manufacturing. So it's applicable to various dosage forms, for instance, standard tablets and hard capsule, but also including the introduction of pellets and mini tablets in hard capsule, mixing powder and tablets in our capsule, but also some advanced technologies such as multi-layer tablets, press-coated tablets. And also we are able to do the packaging, mainly the bottling in HDPE bottles and the blistering, but we are also able to provide powder in sachet. So we have 4 key differentiators. First of all, as we are focused and expert in the oral solid dosage forms, we have developed an enabling oral technologies platform to be able to face the unmet needs in terms of galenic formulation for complex active ingredients. So this one I will explain a little bit more on the next slide. We are also able to deal with high potent molecule, which is more and more the case with recent new chemical entities. That's why 2 years ago we completed the construction of a brand new dedicated manufacturing facility focused on oncological, immunomodulators, and endocrinology products. And as we saw some emerging products needed some small batch size volume in terms of markets, we have also developed at Skyepharma flexible supply chains specifically suited for rare disease products. So digging a bit more in our enabling platform, so we have developed some technology to unlock actually the value of active ingredients, always powered by our formulation expertise. So we have focused of 4 kinds of technology. The first one is our platform to enhance the bioavailability of product, and that is this platform that Valentine will talking more about on the next part. But we have also some technology to have a better controlled delivery of active ingredients, so modified release and targeting technology to enhance actually the ratio of efficacy over toxicity of the active ingredients, but also some suited technology to improve the adherence of the patients, so with pediatric and geriatric forms, but also some multi-layer tablets to be able to have a once-a-day pill instead of 3 times a day to have to take a pill, and also some convenient technologies to improve the efficacy of drugs by combining therapies. So just a few words on our bioavailability enhancement platform. So Manon has addressed a lot of also already characteristic of active ingredients and the difficulty to solubilize them. So we have a standardized approach at Skyepharma. So first we draw a complete full characterization of active ingredients regarding the physico-chemical characteristics. And when it stands for only poorly soluble, meaning BCS class 2 molecules, so as Manon said, there are some different galenic approaches that can be proposed. So particle size reduction, lipid-based tablets or liquid-filled capsules, but also relying on amorphous solid dispersion using hot melt extrusion, for instance. But when the active ingredients accumulate both poor solubility and poor permeability, it's not sufficient. So this active ingredient falls in the scope of the BCS class 4 molecule, and that's why we invested both in the RCPE program NanoFacT, but also on another program at Skyepharma called NanoMix, which stands for lipidic nanoparticle, which are further dried to be formulated into capsules or tablets or powder to reconstitute. So I will now hand over to Valentine, who will tell more what we have completed so far with this technology and where we stand.
Thank you, Vanessa. For our study, we wanted to really compare the both formulation we developed, so solid lipid nanoparticles and nanostructured lipid carriers that, as Manon said, differ by the addition of liquid lipids in the formulation. We wanted to compare the properties in terms of stability, of encapsulation efficiency, and also on oral bioavailability improvements. The model active ingredient that we chose was abiraterone acetate, which is indicated for castration-resistant metastatic prostate cancer and is available on the market as the name of Zytiga. It has many constraints, such as its insolubility in water, its low intestinal permeability, which makes it a BCS class 4. It is highly hydrophobic and also has a significant positive food effect, which means that the patients have to be fasted before and after the intake of the medicine. It's also degraded by the cytochromes and under the influence of the PGPA flux. And all these constraints leads to the necessity of taking a high dose of about 1,000 milligrams per day. So regarding the formulation, we are formulating our nanoparticles by high pressure homogenization. We first have to heat both phases, so with our lipid phase with solid lipid and the liquid lipid for the NLC formulation, and the active ingredients that we have to heat above the melting temperature of the solid lipid. We also have the aqueous phase with the surfactant and water, which we heat at the same temperature. Once both phases are homogeneous, we mix them together with a nitraturax at 11,000 RPM. Also at 70 degrees, and then we put the pre-emulsion in our microfluidizer for 4 cycles at 750 bar. After the 4 cycles, we cool down our emulsion, and we get the lipid nanoparticles, which present a size of 150 nanometers for the SLN and 109 nanometers for the NLCs. We wanted to study the stability of both our nanoparticles in digestive fluids. So we did the study for 2 hours in simulated gastric fluids, in 6 hours in fasted simulated intestinal fluids, and also 6 hours in fed simulated intestinal fluids. We studied the mean diameter of both our systems. So for the SLN, we noticed a high increase in terms of sizes, going from 150 nanometers to 1,000, which means that the SLN has a tendency to aggregate. This aggregation we did not find for the NLCs. So even in fed simulated intestinal fluids, we only had a slight increase of about 300 nanometers, that it's still acceptable. We also studied the liberation profiles of our active ingredients. So in the same simulated fluids, and we noticed that for the SLN, we also have a slight increase of release of the RPI, but only of about 2.5% of the total amount that we put in our formulation. And also for the NLC, a slight increase, but only of about 1%. So all this study allowed us to think that the NLC have a greater impact, a greater protection of the RPI in simulated fluids, both gastric, intestinal, but also in fed fluids. We also made a proof of concept for the enhancement of the oral bioavailability of abiraterone acetate on rats. We had 3 formulations that we administrated. So we had free abiraterone acetate in suspension. Also, we had SLN in suspension and NLC. And what we could get from this study is that we had an improvement of the oral bioavailability in the study, so of about 1.25 folds for SLN and 2.35 folds for NLC in comparison with the control, and a reduction in the variability of the API absorption, and in particularly with the NLCs. To conclude, this study allowed us to prove that NLC are better than SLN. We have a better encapsulation efficiency, a better stability in digestive fluids, and a better improvement of the drug absorption orally. And we are still ongoing. We have an ongoing demonstration of protection against the degradation by the cytochromes, the first pass metabolism, and the efflux pump.
So thank you to all our speakers for those very insightful presentations. As a general conclusion of what we've just seen, so we've seen that the NanoFacT consortium allowed an integrated continuous approach to advance a robust, flexible, and quality by design manufacturing of nanomedicines suitable for industrial implementation. And we've also seen that advancing nanomedicine requires a holistic approach. We've seen a presentation of how excipient science is important, the formulation know-how, and how innovative manufacturing solutions contribute to this approach. With the NanoFacT consortium, we moved closer to delivering nanoparticle-based medicines which are effective, scalable, compliant, and ready for real-world use. So let's continue the conversation. We have 10 minutes before us for the Q&A sessions. So you are welcome to ask them in the questions tab that should appear on your screen. And I will ask them to our speakers. Okay. So maybe a first question for Manon Gattefossé. So which lipids can be used in a solid dosage form for pediatric patients, and what are the maximum amounts used in pediatric patients?
Yes. So for pediatric patients, it's for oral solid dosage form, or it's like for all dosage forms?
It's not precise.
Yeah, okay. So, I think we have like a couple of market reference already that use our lipid excipients for pediatric forms. Comritol, 888 atopressirol, Geleol, I think they have been already used for that. Regarding the amount of the product you need, this is not something I have in mind, but I know that we have done like several presentations on this subject. So this is typically something that we can share with the customer because depending on the excipient, depending on the age of the patient, this is something that needs to be adapted. But we have this data internally, so we can also share if needed.
Yeah, thank you, Manon. We have another questions about encapsulation efficiency, so maybe targeted to Caroline and Valentine. You will tell me who wants to answer. So how did you determine encapsulation efficiencies?
In our case, we separated the aqueous phase from the nanoparticles and then determined the aqueous phase via HPLC. And then we had a formula or an equation that we used to determine the encapsulation efficiency.
Okay. So hope that answers your questions. Another question to you, Caroline. Did you perform comparative stability studies by evaluating the variation of the zeta potential of manufactured LNPs? And if yes, what was the impact of the manufacturing method, bottom-up or top-down, and the type of LNPs?
So in general, for the top-down line, we performed stability studies. For the bottom-up line, we did not focus on stability. So there the focus was the digital framework and the quality control. There we had no stability studies, but for the top-down line, we did stability over 8 weeks.
Okay. Another question that was asked, when working with nanosystems, solid or liquid, how can you detect API release from the particles when 100 nanometer will pass through a syringe filter prior to analysis by UPLC? Don't know who could answer that among the team.
Because we use centrifuge, ultracentrifuge that we filter, and we only dose the surfactant, the supernatant. Yes, sorry.
Yeah. Okay, makes sense. Okay. Another one. How can lipid and surfactant excipients be effectively screened for NLCs to achieve high drug encapsulation efficiency while ensuring that drug release is not adversely hindered by excessive drug lipid affinity?
Can you say that again, Margot? The last.
Yes. How can lipid and surfactant excipients be effectively screened for NLCs to achieve a higher drug encapsulation efficiency?
Okay. So I maybe I'll let Caroline and Valentina answer that because they have performed with active the different formulation, but I guess you need also to get an idea about the solubility of the active into the different lipids, and then also to check if the surfactant that you use is still effective to maintain droplets that are stable. But I don't know, Caroline and Valentin, if you want to add how did you screen the different lipids you wanted for the formulations?
Yeah, for us it was, as you mentioned, you need to determine the solubility of the drug, of course, in the different lipids, and then also determine, especially for NLC, if the liquid lipid and the solid lipid are also miscible. So this is important to determine then, first of all, solubility in both separately, but then also together.
And if I can add also on that, for the solubility of the active into the solid or liquid lipid, we have some data already with some actives internally, but we have also miscibility tables. So this is something also we can share depending of what is needed. And but obviously, when it's a new active, you need to perform the thing into the lab.
Yeah, exactly. Okay, thank you for your answer. Someone has a few questions regarding the top-down approach. So what is the single biggest challenge in the manufacturing process? And in terms of overall production, is the bottom-up approach generally superior? Maybe for you, Caroline.
No, I would not say that bottom-up is generally superior. It depends a lot which type of formulation you want to produce. So with bottom-up approaches, generally you need that your lipid is soluble in the antisolvent. So there you always have an organic solvent, which might not be the best for certain cases. And in top-down, of course, you have high shear. So we work with high pressure, which is for drugs that are more sensitive to high pressure, for example, nucleic acids. This top-down is not the best route there. I would say the bottom-up approach is the better one, but it depends a lot. But maybe Vanessa or Valentin, you also want to say a few words about this.
We cannot hear you, Vanessa, if you are speaking.
Caroline, say it again, because we were looking at another question, actually.
No worries.
You can say it again.
Maybe you can also add something if top-down or bottom-up is the better route to produce nanoparticles.
If it works, sorry?
What is better, considering the manufacturing, bottom-up or top-down, in your opinion?
Okay. So in my opinion, so at Skyepharma, we only have a high-pressure homogenizer system to approach the manufacturing of nanoparticles. But what is great is that it's solvent-free system, so eco-friendly approach. So so we have not experienced the other way except through this NanoFacT consortium. But so we have achieved great stability, and we have now collecting some data that are robust in terms of this nanoparticle, which will be actually for us an in-process intermediate material, because it's supposed to be engaged after in drying process to be further stabilized into oral solid dosage form. So we are confident that this this approach is leading to nanoparticles that are sufficiently stable without using any solvent, with good encapsulation rate. Also, Valentin did not mention, oh yes, but she achieved 100% for abiraterone, which is very good. And so it's this process is particularly suited. So we have two equipment at the industrial scale at Skyepharma, and it's very scalable also, because so the brand we use, microfluidics, and the interaction chamber, it's a scalable process from lab scale up to industrial scale, because you only multiply actually the number of interaction chamber for the scale up, so it maintains the same geometry for the process, so you can expect to have some reliable result at scale up.
Yeah, thank you. And Valentin, about the encapsulation efficiency, we had a follow-up questions regarding what you said about the centrifugation. So did you centrifuge your samples and measure the supernatant, or did you use a filter?
We use a filter that when we centrifuge, and it allows us to measure the supernatant.
Okay.
Yeah, so there are some ultra-free filter with a specific porosity that retain the nanoparticle in the filter, and it only goes the liquid without nanoparticle, and the supernatant is recovered at the bottom of the tube. So it's an ultra-free device.
Yeah. Okay, perfect. So as we approach to the end of the webinar, I see that we have other questions unanswered, but do not worry, we will answer after the webinars by email. And if you missed the beginning of the webinar, also no worries, the recording will be available on our website and our partners' website upon request very soon. And the second session is organized on April the 2nd. You can also register to it. So with that, I would like to thank again our speakers for today's webinar. Thank you all for your attention, and I wish you all a good day. Thank you.
Thank you, Margot. Thank you all.
Thank you. Bye.
Bye.